In addition, bring the test questions homework. That's the last assignment for this class. Wednesday, chance to both begin your studying, since it's something you get to read through and look at. Maybe you choose things that you know are a challenge for you, a little bit more difficult, or ones that you want to make a point of reviewing and get some points for it. There's extra homework points available too if you add additional questions. And then we'll talk about all that stuff Wednesday, and the final exam here next Monday, same time and place. A couple announcements from around the department. I know, right? Now it's December, and things are continuing to move quickly. A couple things around the department. Library books, any things you've checked out from the resource room are due next week. Grades don't get released until you return that. There is our holiday party a week from Thursday at 6. Please come. Friends, family, welcome. And for next term, if you're interested in being a student assistant, like I announced at the beginning of this term for fall, that's you get credits toward your tuition covered by working in the department. The application for that is open now. It's due, I think, a week from today. It's a short single-page application. You can pick it up from the resource room or from the board right outside the room. And make sure you get it in by the deadline that's given on the page. If it's noon, then we're looking at it immediately after that to decide. So that's for winter term, chance to get your credits covered and work with us then. And other announcements, things coming up we could do related here. Registration's open for winter term. Get in there, take a look. Remember, you can always drop things in the first 2 weeks at no cost. So better to get into something and let it go, and make sure you got a spot. And if you got questions about particular courses, ask the specific instructor or our department chair, April, or definitely Kara Leonard, our career coach. As well.
On that note, if you haven't already filled out your FAFSA, it's a really good time. Make sure you do that.
Absolutely. We got year ending here, tax things being completed, opportunity to get yourself ready for next year's aid season. Yeah. So today we'll go through the quiz. Make sure that we're as clear as we can be on photosynthesis and respiration. Talk about the homework environmental responses related to hormones and tropisms, and then talk about plant nutrition. There's a couple very important nuggets to take out of the plant nutrition discussion, and at least a question or two will show up from that in the final exam about how nutrients move in the plant and where we see signs of deficiency, which is another tool in your toolbox. Now you understand how the plant's going to respond to the environment as far as temperature, light, is it making food when it's dark out? And if we add in, now I can notice something wrong and determine maybe not the specific nutrient, but is it mobile or not, gives you a good head start on what to do about it and to make sure that you intervene as quickly as possible to avoid future problems. So I'll get there, but first let's start with the quiz questions. I'm having, I can't get this to transfer to the document cam as usual for some reason. So you got a quiz in front of you, you know what the questions are. Number one was, what's the equation for photosynthesis? And this is not it. It's close.
It's the opposite.
It's got some of the same things in there, right? Yeah, it's the opposite. Carbon dioxide and water with light, right? You gotta have that energy source mentioned somehow. That's the thing that makes this go down, forms that glucose sugars and oxygen release. So again, respiration, and you can just memorize how those look or sound, whether it's, like CO2 plus H2O or carbon dioxide and water, then you can generate a lot of things from that, both respiration the other way and things that come out of it. Number two, asking about one of those things from photosynthesis. It's a gas, that's carbon dioxide. People widely understood this comes from the atmosphere, it gets into stomata, and it shows up in the Calvin cycle of photosynthesis, and the light-independent reactions. Down here, right? This is where that Rubisco, or PEP in C4, chem plants grabs onto carbon dioxide and starts building that glucose. So that's second step. Continuing on with photosynthesis, right? The energy from the sun drives this thing. What's the plant pigment involved? That is chlorophyll. Mentioned way back up here what this stuff is. Occurs in the chloroplasts. That's where we find the chlorophyll. That's doing that absorption of high energy light, both in the blue-violet spectrum with the most energy, so the answer to the last part of the question, and also in the red-orange spectrum, right? And green is what's reflected, so plants are not generating or absorbing energy there, and that's what we see. And these were some of the bonus point answers, different pigments that contribute to absorbing some energy in specific colors in the plant. That's number 3. Stopping here, not clear, you want to go more detail on any one of these. Number 4, the products of the light reaction. One of these is especially important to photosynthesis. And why is that? All here. So the photosynthesis, light reaction, the big part. It excites those electrons in chlorophyll A, and you end up splitting water, you get oxygen out, and then we get that little bit of energy. Right, this here, ATP and ADPH produced, that ends up starting the Calvin cycle. So that's what I was trying to get at there, is the result of the first set of reactions. One of those results is this energy that then kickstarts the next step. So there's this relationship there that you gotta have one to get the next. Number 4. 5 is photorespiration, and how, when does it occur? What is it? How can we avoid it? And right, these two address that. You see that what it is a situation where under hot and dry conditions with stomata closed, we're short on carbon dioxide now. This Rubisco imperfect enzyme to do this thing starts bonding oxygen instead of carbon dioxide, and the result is that we use a little energy and make more CO2 instead of making plant food, right? So it's a net loss for the plant. And our number one response is open up stomata, right, to get that CO2 in. It's the first line of defense. The others, develop a strategy like C4 or CAM plants to either separate in space, right, the process of photosynthesis and do that in the bundle sheath like in the C4 plants, or in time and open at night and save that CO2 for later, like in the CAM plants, are the ways that you can deal with that. Right? Remember this one here, so they've separated out that direct connection with the air and made a big opportunity for photosynthesis in the bundle sheath in C4, and in CAM we know about the nighttime storage. Stomata open only at night, in C4 stomata always is, hold it for you here, that growth is slow because it's got this conversion. It only can absorb at night when stomata are open, and then it has to turn all of that malate back into CO2, so it's taking extra time and energy to do this, which slows things down, but facilitates survival under extreme circumstances. Then we have a section on respiration. Next, question is, name the products, what comes out of it. You know, that it releases water, but it dries things out a little bit, releases carbon dioxide. So we say we exhale carbon dioxide, plants take it in, although they really do both. And energy. That was, the most points on this question was, the focus of this process is getting energy out, right? Turning that stored food molecule, glucose or starch, into something that we can grow with. Which gets to number 8. What happens when plant produces more food in photosynthesis than respiration can use, which is the whole green scenario here, all that's in between, more photosynthesis than respiration. And so what happens? You know, the 2-word answer was it grows or growth. Food is stored, food storage, we put that stuff in, like, yeah, you're getting at it. But the idea here is that then there's the capacity, right, to grow, right, to get bigger beyond that base level metabolism of survival. Number 9, the connection between temperature and these processes, you know, is also right from here, you know, that it goes up to a point, photosynthesis drops sooner, respiration drops later, but eventually they hit a high point where they can't do more. And there's a place where there's no photosynthesis happening, right? Like the opposite of here where it grows, no photosynthesis happening, still relatively high respiration rate because temperature is very high, but not too high to shut it down yet. And that's where we're consuming all of our stored resource, we're running out of energy. That's it. And I was trying to get at that thing in number 10. Why is aerobic respiration more desirable? It makes more energy. We got 16 times more, right, numbers of ATPs, but generally fermentation or anaerobic respiration, low energy, aerobic, once you feed that fire with oxygen, in the last step, then you get that big payout of more energy. And where's this happening? In the cell, that's mitochondria, right? The powerhouse, mighty mitochondria, energy makers in all of our plant and animal cells. And then the bonus was asking you about those. Carotenoids, xanthophylls, anthocyanins, the colors that they produce, and what happens in the Krebs cycle, also known as the second step in respiration, depending on the level of detail. There was some points available here. Basic idea is carbon dioxide is produced there, and a little bit of energy as we continue to break down glucose and get close to that point in the 3rd step, where we're going to get a big energy payoff by incorporating oxygen into the process. So it's right before that. Keep in mind that at the end of the game here, whether we're growing or storing, right, and storage can lead to later growth, only about 40% of what happens in respiration is used for that, and the other 60 is lost. Questions? Nothing yet. If they pop up, ask. Otherwise, I'm gonna take a look at the whole one. some connection there to, if you don't have this thing, then growth is not gonna be as inhibited in some way to these two, and it's gonna be not as promoted, right, in some way to the top three, just in a general sense. So then it's a little bit more specific for each one. Like if we don't have auxin, then we're gonna lose apical dominance. We're gonna lose that rooting. And we're gonna lose that shoot inhibition, right, which is the opposite of cytokinin, which is gonna do root inhibition and encourage shoots. But as we take away one of those, that's gonna shift that balance between the two. So for each hormone, you're looking at exactly what it says here, right? And then thinking about, oh, well, if I don't have the ability to stimulate shoot growth, then what happens? Remember, the antagonistic effect here in this relative quantity of cytokinin and auxin, determining, do we get more roots at this site, do we get more shoots at this site, and how is apical dominance looking? Where is that management from the top, that auxin that is inhibiting the shoots immediately below, right? Because now it's shifted to more auxin than cytokinin, so that says inhibit shoot growth. But if we bring that out, pinch it out, right, then things change again. And that's the question too. What happens in this case? Where does it go? And what kind of response do we expect to see? What do you think? If it's broken, Sal?
I have a question about like what happens when you break the up, like when you basically like you pinch something and like lateral buds start growing. At what point do the lateral buds produce enough auxin to become like, you know, to establish apical dominance basically?
Like after, so once I look at my rough drawing set here. Okay. So this is our apical bud. And you're saying, when we remove that,
Yeah. At what point do those lateral buds now produce enough auxin to become like a, you know, the dominant, the nerve
yes, like,
because it produces more cytokinin, right? So like.
so the first thing that happens is that the auxin, let's say, for the sake of this, right? The auxin is blue, and then there's some here, and there's some here, and we run out. So when this is gone, now this is just shifted to, because there's less auxin, the effective cytokinin is raised just by removing the other thing, right? Because it's a relative relationship, how much of one, how much of the other. So we both stop that inhibition and get cytokinin's effect of promoting those shoots to grow. And then, yes, but it's like a time frame of, it's not just immediately this quantity shows up here, right? Because that's physically removed if you cut it off.
Right.
So it takes, 24 or 72 hours to regenerate
Okay.
more to become like the new apical meristem, right? The new bud that then has this higher quantity, which then sends some of that to be here, and some of that to be here, right? And then maybe that.
Got it.
And so initially, then we get that side branching shoot development, right? Which is like when you pinch something, you're thinking you want to make it bushy, okay? You want to get that result. And then this is going to have it, right? And this, maybe, not quite sure, depends, right? How much light is available from the side versus the top, and et cetera. But the general decreasing quantity of auxin relationship continues, but not immediately. There's some time there where now these nodes are going to start making it more, but in the meantime, still all of that is lost, and so that shifts this relationship to, you get the upregulated effect of, positive effect of cytokinin rather than the inhibition effect of the auxin. So that's what we expect. See there is lateral shoot growth at the nodes below, and then eventually inhibition further down. And number three, the flashing light during a long night, have a short day. We spent a bunch of time last week talking about this effect, and I said you gotta talk about PR, PFR here. Remember. So, two things at play here. One is the biological clock. Sunlight quickly changes the red light absorbing thing, which red light happens during the day, far red light, FR, happens at night. So the red light thing during the day quickly transitions to PFR, and PFR over night slowly in darkness measures dark period for that flowering control signal. And for that short day plant with the light flash, remember it divides that long night into two, making it now short nights and repressing that flowering signal. This is the example in question 3 that's being asked about. You do that, and you're splitting that into 2 short nights. And of course, in the opposite, where short night is what triggers flowering, when you create that, you're inducing that signal.
Quick question on that.
Yeah.
In a practical setting where a greenhouse is trying to do this, or like, how long an exposure do you need to raise the PF here?
Yeah, there's a little bit of a relationship with what the light source is. Like if it's fluorescent lights, you're gonna need a longer exposure. If it's, high energy LEDs, like the most modern greenhouse lighting, you just need a period of 5 minutes, 10 minutes, 15 minutes to change that effect. As long as, initially, you gotta experiment a little bit, because it depends on how many of the lights, are they reaching all the plants, how far away, the distance overhead has some impact, but generally a short exposure is all you need. Because that short exposure still makes still changes my effect and say, oh, there was a lot of light. And so at nighttime, you need long exposure to revert back to the DR. But all you need is a couple minutes of absorption to shift that into PFR and say, hey, I recognize that there was daylight out there. And that's why, part of why the plant is, the important time that it's measuring is the dark, because it's easier to mess with the light.
What is far red?
Far red.
Or like, how would that be,
What's that?
how would that be defined?
Far red light. So that's like the edge of red and infrared light here in the visible light spectrum. So red and then far red is like the edge here. And it, once you're to the left of it, that's technically infrared. But right around here is you're, yeah, I mean, not really disrupting as much as the plant is just sensing what's happening. And so what's happening now is there's light present, which changes that form quickly. Right? Whereas before, the light flash, it was just slowly, anything that was accumulated during the day was reverting back here. And so whatever had accumulated as PR at that point of the light flash, right, the short exposure, just, on that just couple minutes, comes back over here. So now it's just sensing that signal as a day happened.
Oh, okay.
So you're not like interrupting it per se, it's just sensing, just observing phenomena. And what's observing there is the presence of light that changes that form quickly.
And so then is it jumping back into that cycle? It's jumping right back into that cycle then? Right
Yeah.
after the-
Yeah, so this is, this happens almost instantaneously, and then say your exposure was 10 minutes, you turn it off, now it's gonna slowly go this way again, and it's going to measure that next instance as a short night from that flash, until the next light, which might be like the actual morning comes and sun rises and it senses now there's light again.
Gotcha.
Yeah,
Where is
so-
that process taking place? Is that in the chloroplast? Okay, yeah, I was just curious.
And before was advantages of thigmotropic response. Here. Basically, why does it pay off for plants to know where they are in space? Roots move away from something, that's not going to have water or nutrients, that's a good thing. Twining vines have something to grab onto. Navigating the physical world without visual input, it's all about contact, touch, and so it's useful adaptation. And finally, draw this plant. I always find a way to cram something else in there, herbaceous, angiosperm, alternate, simple, etc. And then use the arrows to indicate where these hormones come from. This came out of the reading. I'm not going to draw it all there, but I encourage you to draw the question, answer the question, and draw that if you haven't already. There are a memorable description of all the things that these hormones do, where they upregulate something, gibberellin, speed up our flowering time and our fruit development, for example, but it doesn't have anything to do with losing leaves or encouraging seed dormancy. It actually encourages germination. Yeah, just a quick way to compare. Yeah.
So it's always going to be in opposition. Like there's never it has nothing to do with this thing, it's always either upregulates or downregulates.
No. So in the case of gibberellin, when its seed is exposed to it, it's upregulating germination starting. But it doesn't encourage dormancy and it doesn't stop dormancy, but it does start germination, right? So it works against dormancy, but it's by actually encouraging that growth rather than inhibiting this process. And that's different than auxin and cytokinin, where auxin does inhibit shoot growth. So it just depends on the one. Okay. And then I want to mention this, the phototropism following the sun, speaking of auxin. And talk about this. Make sure that it makes sense that for whatever reason, auxin migrates away from the direct sun. And so that fact, that response to the light creates this whole curving, bending toward the light phenomenon, whereby all this hormone now moves over to the dark side of the shoot tip. It's a growth promoter. Cells elongate there on the side where the hormone is present, and that makes bigger, heavier, more things that push the other side down. So it looks like a bending toward, but it's really being forced that way by just more mass on the other side. Other questions there? Yeah.
Back to hormones, I'm pretty clear on the auxin and the cytokinin. Cytokinin are produced, but not the other hormones.
So gibberellin is produced throughout the plant. So it has several roles. Right, from germination, fruit, flowering, speeding up. So that can be produced at nodes all over.
Okay, that was my question, if it's just at like, where there's meristematic tissue, that's where it has to.
Yep, yep. And, well, you can have cells that are, their determined job or part of it is to produce hormones, but they still have to be produced at that meristem and then.
Are they kind of gonna be co-located within those cells?
Yeah, and they can be moved, from that site a little bit, but had to start there at least. And what about the others? Inhibitors. Anyone know where ethylene's produced? Yeah, where in the plant? Senescence of leaves. Anywhere there's an injury, yes. Leaves generally, epidermis of leaves. It's not the site of production, but it has an effect there. Yep, so mostly connected to its, what we say the functions are there. So in the canopy generally, in the leaves, N-oxalic acid. Likewise, all around the plant, but especially in leaves because of that constant need for control of guard cells. Other questions, thoughts there? All right, well, let's take 5-ish. Come back at 48 or something, and I'll start on planning.
Yeah, so it's just resetting.
So we have circuit, I don't know.
Yeah, or flip side, yeah.
No, I
The presentation's there.
I did take the equipment safety class, so.
This is online, right?
It's online, and
But then there's two in-person ones that you can choose from. My grandparents live here.
they usually have two
My
other.
mom lives here half time.
Then my husband and I.
But there one's online. Sorry, like not very, words of very true. There's two different times, right?
Oh, it just says if you have to take the test. I looked up more, like, through this at all, like, stuff about,
I don't know why I'm home, I don't know from the computer, I had to write down the things that I was thinking. I would have done maps.
but, yeah.
Just like trying to figure out what's about it, and then copy. If you're afraid of that.
Yeah.
Once the online one is allowed, yeah, it's weird, I don't really, I haven't finished registering, so you know, stuff like that. But for the CSA, if it helps, something that I've been doing for that one, got started. And I just use the same template format, you know, that I use for yeah, it's a lot like the spring finals, so that's, like, I didn't want to because it is different, but I started by copying everything and then just like got rid of the stuff that wasn't relevant. Sections and, yeah, and so then it kind of just turns into, you can reuse some of the same clocks and props. Yeah. Yeah, I've been repurposing it for that one together, so. Yeah.
Plug it in and see what I can do.
Are you doing? Anything next?
That's what it's sort of stuff.
Yeah, I did a couple of things. Class and class, I'm still pretty behind. Mostly caught up in this class and I was trying to make a little things. I have a couple of these
the VA or whatever.
Oh, yeah. Well, actually, during the time of the school year,
No, it does for real. Yes, it does.
I'm sure. Yeah? Yeah, that's how we get summertime extra. The college school year is fall, winter, spring, summer's not included. Yeah, so it's split out, as 4 ways if I take summer classes, as long as I don't take summer classes. Because they say that they consider school years, 3 semesters, before it's extra.
That's it.
So Plant Nutrition, as promised, this is the last new material in here, and I just want to get at, how many taken soils already? Soils, good number, okay, so hopefully this is review then, but worthwhile to do so, and to put it together with what we've talked about thus far in this class. Quick reminder, plants have 17 necessary nutrients. The ones needed in high amounts are called the macros. This is them in descending order: nitrogen in the highest quantity, phosphorus, potassium, sulfur, magnesium, and calcium are our plant macronutrients. So, plant nutrition involves the uptake from the environment of all the raw materials required for essential biochemical processes. Just like I was saying back in week 2 about roots, this all comes dissolved in solution in water from the soil where those nutrients are held to get in, unless you're providing that, right? Of course, in a controlled environment, you could foliar feed, you could soil drench, but it's got to get in somehow. Majority of the time, it's via roots in water. Finish up the quote here. Distribution of these materials within the plant and their utilization in metabolism and growth. Right? So these are the mineral nutrients outside of carbon, hydrogen, oxygen that we know are coming in water, carbon dioxide, right, to complete photosynthesis and respiration that are used, mineral nutrients that are used for protein synthesis, right, making the chloroplast, right, that does all of those things that do photosynthesis, right, making the mitochondria that does all of those things to extract energy from glucose in respiration. So now this is like the raw construction materials to actually build plant parts and do plant processes, right, by having enough of the raw materials for the project that you work on. They're needed in the life cycle of the plant, as I was just saying, part of any molecule or constituent essential to the plant. And efficiency symptoms appear when the quantity is insufficient, which tends to be when growth is rapid, which has a connection to times when temperature is in that optimal range, right? Sunlight is prevalent enough, and water, sufficient water. All of the 3 main ingredients help us to grow quickly, and that's when demand for the mineral nutrients is higher. We're growing. So here they are. I mentioned how carbon, hydrogen, and oxygen, right, are actually, those together are carbohydrate, right, that comes from the atmosphere, and the others are mineral nutrients.
Sorry, which, the first 3 come.
Carbon, hydrogen, oxygen, yeah. So they come in as water and carbon dioxide. So this is a look. I'm not going to test you on the relative quantities or anything like that, but just to get a perspective on, like we say, oh, plants need NPK, and those tend to be the ones on the fertilizer labels because they're needed in large quantity and they're harder to get from the soil So you can see, like, this is a relative sense. Overall dry weight of the plant is 1.5% nitrogen and 45% oxygen and carbon. But, so 1 million molecules of nitrogen where it only needs 60,000 of phosphorus, so that's like 15, 16 times less, right? So even in the macros, there's a wide range. And then you can compare the micronutrients, chlorine, iron, boron, manganese, zinc, copper, nickel, and molybdenum, right? And some of those are now, you need 1 million times more nitrogen than molybdenum. So things in very small amount are easy to amend and have enough. Things used in large amount, you need to provide more regularly or in bigger quantities. Things, right, hydrogen, carbon, and oxygen, of course, you need to be getting that in all the time. That water in through the roots. Oxygen, carbon dioxide in through open stomata. So there's a relative, right, big difference in how much of things we need. Part of this is to say that down here, for example, for outdoor growth here, Willamette Valley, Western Oregon soils are generally boron deficient. And right, we just need a small amount relative to other nutrients. So we can quickly go from deficiency to toxicity in the case of boron by putting on too much. So everything here, small additions, oftentimes it's in some kind of mineral that contains multiples, and then take a break so they don't build up. Here we tend to see, right, more movement through the soil, loss especially of nitrogen in the rainy season, and often need replenishing. So, again, to that point from week 2 about the roots and the importance of sufficient water, because there's that whole relationship to respiration, transpiration, that pull that gets water in the first place, manipulating that concentration gradient across the soil profile with the root hairs, right, to get water with those nutrients in easily, right, connects us all the way back. So, as an example, clay particles, and this is true for particles of silt, right, particles of sand, although as the size goes up and the texture, right, changes from silt and clay to sand, it holds less nutrients. Clay holds the most, along with organic matter component. It tends to have negative sites on the clay particle that bond to positive ions, which is convenient because a lot of the main things plants need, right, are positive. And so those are there, and as water moves through, water that's both proximal to the root hairs and the soil, those things are picked up, and as the root does its work, right, of absorbing, then those things are moved inside the plant for actual growth.
So the root itself is doing the work to free the nutrients from the clay? Is that what you're saying?
Though I'd say water is doing the first work of pulling it off of the soil particles, and then the root is doing the work of getting now that like charged water into the root, inside the root. As if there was no, in a dry soil, no water, or no water that can be taken up by the plant because it's all bonded so strongly to the soil particles. The root can't pull it away. There's not enough force there, enough energy to pull it away without it being in water. If it's not dissolved in water, then it's not accessible.
So it's like you're switching out one cation for the hydrogen? For example, like magnesium and the hydrogen will like switch places, and that's how? Yeah.
In some cases, so if you're talking about, say, this hydrogen here,
Yeah.
right, that's gonna go into the water and end up in the plant. Also, right, from all those hydrogen ions that are needed, right, from the previous slide, and then another positively charged cation can get in there. It doesn't have to be necessarily a swap out, but it is like a fill all the bonding sites. And so that's happening, right? Particle after particle after particle, a tiny clay particle, and enough to hold this quantity that can actually support plant growth then. Yeah,
No, this is going on most of the most of it.
as much as possible, that's the goal. Yep. So whenever the plant has that ability, it's doing it.
You are supposed to,
Yeah.
then if not, then it would have to use some form of.
Yeah, sometimes, right, there's going to be a nutrient, maybe it's one of the micros, it's only needed in a small amount, it's worth the energy now to actively transport it in and make sure that, hey, we can form that molecule. For example, magnesium is the center of the chlorophyll molecule. So if you're deficient in magnesium, you're not making chlorophyll, so you're not absorbing, right, all the sunlight you can get. So that's one that, like, the plant's going to target even at an energy expense to make sure, right, it's present. Back on the ideal soil composition, again, this is for in the ground, it's gonna be different in a greenhouse. But the point is the same, whether you're using soilless media or soil, that you need to account for air and water space in the media so that that uptake from the last slide can happen. Right? Organic matter can help with that, right? In outdoor soil, a lot of it comes from the parent rock material, but when we talk about soil, right, it's not just rock, it's the air and water contained in the spaces in between that gives the nutrients in water for roots to grow, right, in the air space for roots to grow in and also not suffocate and rot out down there. Julian?
So that is basically, that 25% is basically the space for water. Like, because the same water is equal in soil. Soil is like the ideal soil has.
Yeah, so if I can roughly draw the, not to scale, here's some soil particles. Let's say this is the extent of it. The water that's held around these and the air in between, because there's some spots where there's enough space where if there's water there, it's going to fall through, it's going to percolate down. So that ideally is about, half of the total profile is airspace that can be filled with water, but also has some air. Because if that 50% is all water, then there's no oxygen available for the roots, and everything is, right, then it's standing water, totally saturated. So we still want available airspace and available water so that the roots can actually contact that and take it up. Just a reminder, air and water are components of soil. So in terms of plant nutrition monitoring for whatever growth situation you're in, we're focused on preventing deficiency or toxicity from happening, because if you're noticing it, then it's already in process, and that's slowing down plant growth, potentially pushing it into the danger zone of, more suffering or maybe death. And so there's a few things that we can do. In greenhouse growing, nursery growing, containerized anything, you can monitor your pH and your EC or electrical conductivity. Have you guys done that here with Frank or others using a little pH EC meter?
It does
Yeah.
that in greenhouse one.
Greenhouse. Right, and so that's a measure of the salts present in solution, right? Again, when you put that in there, it has to be moist, right? You can't really measure it off a dry soil because you don't get a sense of what's actually present. So salts is the proxy there for the quantity of nutrition that's available. So the good thing about EC is it tells us, hey, we got sufficient nutrition here. There's a range for different plants and different conditions. Or it tells us, oh, our salts are too high, that might start to have a negative impact. The downside is that, so EC, salts roughly equals nutrients in this case. It's a measure of what's present. So that's a plus, gives us an idea of what's there. But the negative is, what's that? We don't know the amount of any specific one. Yeah, it's non-specific. So it might be that we got a whole bunch of sodium and not enough potassium, but we don't know without a more specific test.
So I was having a big conversation about these EC meters, and somebody was telling me that they only measure nitrogen. From my understanding, they measure everything.
Total salts, total dissolved salts. You can do other tests to measure one nutrient, but it's usually not dip in the meter. You sent to a lab, they
Because
burn it. and
what it
tell you.
was we were doing we were feeding nutrients, and they were telling me that when I mix the nutrients, that when I put the EC meter in there, it's only telling me the nitrogen, or vice versa, the bag's only telling me.
Unless the nutrients you're using are only nitrogen,
Yes, I was right.
it's measuring all the available salts.
Yes.
Yeah.
Tell them that they know their time.
Yeah, I think that often happens. Yeah. So it's measuring salts, but he is right. It's non-specific in that it's not saying, hey, you're short on potassium, you got good on phosphorus, etc. Right? It's just a general number of like, am I in the range where things seem okay? And of course, pH, likewise, tells you something about, are the nutrients that are present absorbable? Can they be taken up at a given pH? And so the combination of those two gives you a better idea than just one or the other about what's actually happening. So pH of 7 is neutral on the 0 to 14 scale. When we're growing plants, we're generally slightly acidic, so slightly below 7. For field growing, we target like 6.0 to 7 or so. And here's what I'm talking about. Measure dissolved salts. Here's some examples. That's not right. That's not all of them, but it's examples of ones that are there. So salinity, right? Sometimes it can definitely tell you if that's too high, but it doesn't give you specifics on any individual nutrient. If your EC number is too high, one thing you can definitely do is use pure water to flush that out and reduce the total. This is common in constant liquid feed, greenhouse growing. Too high negatively affects growth, and you start to see fluorosis and necrosis. And too low, right? You see some similar results actually. So on both cases, you might notice something is wrong, but then doing the EC meter test can tell you the likelihood of, is that a deficiency problem? Maybe it's too much of something that's displacing another nutrient that you need. Yeah.
So, the role of water, if it's leaching salts out of water, then it lowers the electrical conductivity, but then it says water adds to electrical conductivity, or is it water as a carrier for salts?
Well, water with stuff dissolved in it. So most water, coming out of the tap, say, has some calcium in it, or chlorine in it. So those are going to be red in the test then if you're using tap water.
Okay, got it.
So like if you test tap water, it's not gonna say 0, it's gonna have an EC of like maybe 50 or less.
Okay, so it's not like H2O, it's water.
Water or whatever.
That has things in it.
Yeah.
Okay.
And so distilled water means all those things have been taken out, so it's just H2O, and that would be the most effective leaching agent there. Although if you put enough water, even with some salts on it, it's still going to take things out of solution. connect that to this, here's kind of the optimal plant growth zone, again, 6 to 7, and this is the width, the height of this bar for each nutrient is a relative accessibility of that nutrient at a given pH. So as mentioned in boron, that whole range very available, but if you get really low pH, you start not to see it. Oftentimes, plants growing outside here that are subject to temperature, we're a little bit on the acidic side naturally here. We see phosphorus problems, especially early season when temperature is cold and field is wet. The plants have a harder time taking it up, even if it's present, due to the combo of the cold temperature and the low pH. So if you know you're right at 6.5, you're doing really well on all these necessary nutrients, and so maybe everything is accessible. That tells you that at least that's not a problem. Now maybe you use your EC number to get you closer to what is going on. So there's compounding benefit from having both pieces of information.
Does low temperature affect the uptake of only phosphorus, or is that just?
Especially phosphorus, but so that is about how phosphorus moves in the soil when it's cold. But plants, when it's cold and they're metabolizing more slowly, right? Respiration rate has slowed down. Then everything is getting taken up more slowly. So early season or late season, that's always the case. But in phosphorus in particular, it's more than just like if the plant speeds up, phosphorus availability is still contingent on what that soil temp is.
So with a higher air temp, the plant wants to respirate faster, and the soil temp hasn't come up like it's April or something. It's gonna
Yeah.
cause deficiencies.
Yeah, and then you see those purple leaves on your brassicas.
You could really remedy that aside from like foliar feed of phosphorus.
Foliar food phosphorus is definitely a way, get more in there.
You're not gonna change the soil temp.
Well, you can raise the soil, you can raise the temp in the root zone to, say, cover with clear plastic, a couple of sunny days, right? I mean, you gotta get the right conditions, but if you do that for a week before you plant, then you simultaneously germinate all the weeds under there, and, maybe you warm the soil up 3 degrees, which is not nothing. But yeah, it's slow change for sure. So the best things to do are either wait if you can, or make sure that you supplementally.
Keep them in the medium inside the greenhouse where you can supply temperature and nutrients and control the space.
Yeah, I mean, in the best case scenario. And sometimes, like, if you know from soil tests, let's say, or you just fertilized, phosphorus is present, then you just have to wait, and then maybe in 2 weeks, it's not like the plant will die, it's just gonna show that there's not enough phosphorus, usually that's purpley leaves, which photos are coming up on that. But maybe in 2 weeks now before it was mid-April and now it's first week of May, if it's soil's 5 degrees warmer, you're gonna see a change. It's gonna get greener. So to that end, row cover, little plastic cover, anything that makes it warmer serves that purpose. So speaking about observation, this is really the core of the useful nugget on this plant nutrition. Is okay, so it has all these nutrients, there's different amounts, etc. But what we really do with that information is observe, right? And we use our pH and EC context to tell us something else. And so these plant essential nutrients are divided into 2 major categories, mobile and immobile. And here, I'm talking about in the plant. So, plant mobility. Before we've mentioned things like nitrogen washes out of the soil when it's wet, right? And that's soil mobility. Right? Nitrogen is very mobile in the soil, phosphorus not mobile in the soil. Right? But this is how things move inside the plant or don't. And there's basically, there's the mobile category and immobile. So the mobile category, and here I've outlined conveniently for plants, right? Many of our macronutrients fall into this category. So NPK, magnesium. Are mobile, meaning when the plant is running low on any one of those, it can use more of it, more nitrogen, phosphorus, potassium, or magnesium from elsewhere in the plant. And so as a plant's developing, it's putting on new growth, right? Whether that's tips or side shoots or flowers, whatever. And as it's developing, it says, oh, I'm short on nitrogen. I'm not going to be able to make this new set of buds without it. Then it takes from the oldest leaves down below. Now they're maybe shaded out. They've kind of served their purpose. And they're less essential than this new growth that's going to get us to flowering or fruit production, or just continuing the life cycle. So literally taken from one spot and put into the new growth for further plant development. So that's mobile. Right? So taken from the older leaves to the newer. And thus, we see those deficiency symptoms show up in the older leaves. So you see yellowing on the low leaves. That points us toward one of these and not one of these. Right? Because we know that it had to be mobile in order to be stripped away from the old leaves and incorporated into the new. In contrast, the immobile nutrients can't be moved around because of their size, their shape, the energy input, and the level of complexity that would be needed to shove them into the flow and get them going. They're built into structure, relatively semi-permanent. So just take calcium as the first example. Right? The calcium that's incorporated into those same older leaves can't be removed and put into the new construction, right, of the new leaves coming up. And so instead, we see that in the new leaves right away as they're developing, because they're making a thing, right? They're creating something without one of the supplies, right? They're short on this material for the leaf, for the new flower. And so that means you might end up with a deformity, you might end up with just some chlorosis, that yellowing symptom in the new leaf. And it's risky then, because this is the future, right, of your life as a plant. And so if you can't get those things from somewhere else and you don't have them, then you're developing incompletely, right? And that could be a risk to reproduction. So immobile nutrients we see in the lower leaves first, in the newer leaves first, because they can't come from the lower leaves. Right? So we see them directly at the site where they're trying to get used, but they're not present enough. Calcium, iron, manganese, zinc, copper, boron, sulfur. So most of the micronutrients, with the exception of calcium and sulfur, fall into this category. So that's the major takeaway here, is mobile nutrients, deficiency symptoms noticed in the older leaves, immobile nutrients in the newer leaves. Of course, if you're providing enough nutrition, sufficient water for that to get in, temperatures that allow for growth, then you can avoid this altogether. Right? But things are generally not perfect, and this is our first step in observing and understanding what we see. Is it in the old leaves or is it in the new leaves? Or sometimes it's everywhere all at once. Then there's also toxicity. Some things that having too much of them is also negative for the plant. Ammonium, iron, manganese, boron I mentioned. In both cases, deficiency or toxicity, the first things we notice are chlorosis, that's yellowing, right, which leads to necrosis, that's actual death, so yellow to brown as the tissue dies. And we talk about where we see this. Is it on the leaf margin? Is it in the veins? Is it between the veins? Where do these symptoms exist? So we could say, oh, it's in the new leaves and it's between the veins, or it's in the old leaves and it's on the leaf tips, for example. So both the type of leaf, new or old, and the specific location on the leaf, back to our what's the margin, what's the vein, right? Gives us some clues about what the problem might be. Here's info on some of the main ones. I just added this in from a textbook that I like. If you want to read more, it describes in writing some of the deficiency symptoms. For example, with nitrogen, what's the function? It's in amino acids, proteins, chlorophyll, etc. And what we see in deficiency is chlorosis, especially in the older leaves, because it's a mobile nutrient, right? So sometimes they become yellow and then tan as they die. So it just gives you a little bit of an idea. Of course, I think it's most useful to actually look at some photos and see what's there. This is just a continuation for the micronutrients. As you're making the decision about how to add nutrition, as we were kind of getting at it with like the foliar feed, how do we deal with that phosphorus issue, pay attention to the label on whatever you're using. And you know that the big 3 NPK numbers up there, but for example, this 2010-20 product also provides some magnesium, some boron, some copper, some iron, some manganese, some molybdenum, and zinc. So maybe great if you need a lot of those micronutrients, but if you're already borderline for iron toxicity, then you need to choose a product that's not increasing that iron total. So paying attention to the whole label, not just that 3 number across the top, is essential to make sure you're making the right call on what you need and what goes in. And so now some photos to try to get the picture. Nitrogen deficiency, you know, again, I showed how this is the macronutrient needed in the highest quantity after carbon, hydrogen, and oxygen. So it's usually a good place to start when you see yellowing, is suspect nitrogen, especially if you notice it in the older leaves, right, which is the site of our mobile nutrient deficiency. Here's like fully nitrogen deficient, and here is, right, you can see bigger, older leaves. It tends to be in between the veins. Veins are still green because as the nitrogen is moved, right, it can be dumped out of the vein into smaller and smaller vein and get to the leaf tissues that are out there. So those run out first, the furthest away from the midrib, and then the midrib stays green until eventually, if there was no more nitrogen available, right, that's all going to be yellow. But we see the transition happen in between the veins first. So that would be intraveinal chlorosis of the older leaves, indicating nitrogen deficiency. Phosphorus, we're getting at this a little bit. It tends to be noticed at the tips. Oftentimes there's a mark like this, like an initial, and then some purpling along the margin. Same thing here, this is tomato, and we see the purpling at the veins in this case for phosphorus, which is also mobile, right? So you'd expect to see these things in the old leaves first. And potassium, I'm going through a big NPK, they all happen to be plant mobile, which is great. Oftentimes the potassium we see also at the leaf tip and the margin like phosphorus, but starting in little dots, little points, right, where there wasn't enough potassium to make that tissue. And then as those individual little points fuse together, you start to see more blobby effect all along the margin. And often in between the veins first. So again here, I'm not gonna like test you on, is this potassium or is this phosphorus deficiency? But I absolutely do want you to understand the difference between where a mobile nutrient's gonna show up and where an immobile one.
So is that just a rule of thumb when you're trying to figure out what the deficiency is that if the veins are still green, then it is a mobile nutrient that is deficient?
Generally, yes. So your kind of your, because if it can be moved, it's gonna be at least going through those veins, right? And so the furthest removed from the midrib, the big veins, is going to be noticed first. So you observe, it's kind of like the cascade for developing your deficiency. Symptom observation, observe, and then you're thinking about, is it old or new? And then in either case, say like, what's the location? That is to say, is it veins or not, margin, etc. And then what's the damage? Oftentimes it starts with fluorosis, and then each layer gets you closer to some answer as you're ruling things out. So if you come this way, your first thing is, oh, I see it clearly in the old leaves and not in the new, and then you've eliminated all those things that were over here: calcium, manganese, sulfur, etc. Speaking of calcium, this is a common one. It's macronutrients, it's needed in quantities, which macros are more likely to be deficient for solely that reason. And also, it's not very mobile in the soil or in the plant. So getting calcium level right depends on good fertilization, steady watering, and you can see here, in this citrus, new leaf, right by where fruit has formed recently, we notice that on the margin here, this big bleached out section needs calcium. Here in strawberry, you see it a little bit differently. It tends to show up here at the tip, and quickly turn to necrotic tissue, which could look a little bit like this, but instead of going red, purple, and creeping in the case of phosphorus, which you would expect to see early season on strawberries, because they're flowering in March and April when it's still cool soil here. It doesn't continue on. It just, that area, because it's new leaf, that tried to form when there wasn't enough calcium, didn't happen. Right? This stuff already formed, and it worked, and then it ran out by the time it got here. So it's not like spreading in that way. It just hit a wall, and then it couldn't use anymore because it wasn't there. So this is where we shifted into an immobile nutrient example. Magnesium, similarly, see it in the new leaves, right? All these tops, it tends to be widespread. Well, it doesn't immediately appear in the veins, which would kind of complicate our theory. At the same time, it does fill the whole thing in eventually. And sulfur, I think sulfur's quite tricky in comparison, to be sure that what you're looking at is sulfur deficiency, because while it's technically immobile in the plant and the soil, it sort of shows up everywhere all at once, which I hinted at a few minutes ago. So you see it somewhat in the old leaves and somewhat in the new leaves in the same way throughout the plant, because of the way that it's used, here, right, not enough sulfur in this example, you can see it's basically yellowed, right, throughout. So that one can be mistaken for nitrogen relatively easily, but again, I'd say start your suspect list with nitrogen, double check, is a new growth seem deeper green? If there's a clear difference, then you can rule out sulfur. And I just want to make the point about toxicity too, because oftentimes it's not having enough, right, being deficient in something. But there are instances of too much, and iron is one that goes both ways. Soils here are usually quite high in iron, puts us on the edge of toxicity. So it's immobile, which means we see it in the younger leaves first. This is what typical iron deficiency looks like. It's just kind of striping of dark green within the yellowing between the veins. And this, sorry, the lighting makes it not very good, but it's a bunch of dark splotches, like necrotic little points that then fuse together into bigger necrotic spots in the toxicity example. So instead of like kind of a, you know, stripe of it here, it's these individual dots that fuse together on the toxicity side.
Also new, oh, toxicity.
What's that?
Toxicity is also the new.
Yes. Yep, because in both cases, the iron is being incorporated at the site of development and not mobile. So if there's too much there, then you can get this dark growth, which, you know, it's dark green leaf under there. It's not horrible, but too much iron is the risk with toxicities is the displacement of something else. So if iron exists mostly in plants in this form of a 2+ cation, then other things that also are a 2+, which we saw like magnesium was, for example, calcium was, for example, then it might be displacing that other places. So having too much iron also means that it might look like you have not enough of magnesium or calcium, when in truth, the soil could have plenty, but all those spots in the plant are just being filled by iron, which looks, you know, effectively when things meet, seems the same. Boron, I mentioned boron deficiency, common here, especially in the fruit stage of plants as they're reproducing. It tends to have a higher boron requirement at flowering and fruit production, and we see this like typical cracking sort of, or inside the flesh, blotchiness, that, or this like lumpy texture on the outside, clues about boron deficiency. Again, determining those things is a task for another class. There's like tree care diagnostics, for example.
Oh, I was just gonna ask a question about boron.
Yeah, go ahead.
What causes error? Am I right?
The crack.
Yeah.
Yeah. So like the photo on the right. Yeah. It's so as this flesh is forming, right? It needs just like this tiny, just a couple molecules of boron to correctly make the next piece of like flesh in the fruit, and then it doesn't have it. But it seems like here what's happening is it also has too much water, so it's both not forming in like what could have been like just a more narrow slit, and then as the water pushes it, then it moves to look like this bigger crack.
So it's kind of like if it's low at any point, it's most likely gonna ruin the whole.
At least during fruit production. Yeah. Okay, and so here, I was trying to put these concepts together into kind of a closing synthesis simple guide, right, making that division between new growth and old, where we, right, old, we see typically nitrogen, phosphorus, potassium, and calcium and iron, two of the primaries that we see in the new growth, just as a way to make sense of it all in one place. So again, a couple words on a page is not the definitive diagnostic chart, but the reasoning behind how things end up in one place or the other is consistent. And that's what I get to say about plant nutrition and how it connects to our basic plant science functions from the other 9 weeks. Questions there so far?
When it's not mobilizing the carbon dioxide, is that probably it's just not there in the environment, and it's just like a indoor grow or something, because in normal atmosphere, there's probably not likely to see that.
Open those stomata, get some more, yeah. Yeah, usually that's in a situation where you're running a greenhouse as hot as you can, and so plants are starting to shut stomata for water loss, and then you get to photorespiration, maybe not with CO2, but yeah. Generally, it's the mineral